Hello everyone, my name is Meghana Pagadala and I'm an MD PhD student at UCSD, co-mentored by Drs. Hannah Carter and Jill Meseroff, and today I'll be presenting my talk titled Germline Variants that Influence the Tumor Immune Microenvironment Also Drive Response to Immunotherapy. Cancer is an extremely complex and heterogeneous disease, but it is also heritable. Twin studies that have been conducted estimate the heritability of cancer to be approximately 33%, with some cancer types such as melanoma and prostate cancer having heritable components greater than 50%. However, risk loci that have been identified through Genome-Wide association studies to date only explain a small proportion of this heritability, leaving a large proportion poorly explained. Thus, my research is focused on better understanding how host genetics interact with somatic phenotypes, in the hopes that this can explain some of the missing heritability. The main difference between germline and somatic variation is that germline variation is inherited and present in all cells, including immune cells, where somatic variation is acquired during an individual's lifetime and present only in affected cells. But both types of variation can play a role in tumor initiation and progression. Another critical component to tumor behavior is the immune response, and numerous studies have shown that germline variation plays a large role in immune traits. However, currently we don't have a good understanding of how germline variation modifies tumor immune interactions. And furthermore, germline variants are present in tumor cells, but also in immune cells. But we don't have a good understanding of the cell type effects of germline variants in this context, and whether germline variants may be acting more strongly in immune cells compared to tumor cells or vice versa. So the goal of our study was to understand germline variants underlying the tumor immune microenvironment, and we did this by doing a discovery analysis with 137 heritable immune phenotype or IP components in the Tcga, and then integrating databases such as DICE and GTEC to gain a mechanistic understanding of these variants and their cell type effects, and then lastly to evaluate the role of these variants in immunotherapy response. So the first order of business was to characterize the tumor immune microenvironment in the Tcga. So we started by collecting 741 immune phenotype components or IP components from literature, and this included 724 immune-related genes, such as genes in the HLA region, antigen presentation pathway, TGF-beta signaling pathway, and more, and 17 composite phenotypes, including immune infiltration calculated with CyberSort-X, and also immune states for Thorson et al. We then wanted to determine IP components where a large proportion of phenotypic variation was driven by genotypic variation. And so we conducted genome-wide complex trait analysis or GCTA analysis and identified 137 of these heritable IP components shown in the upper right here. And so a number of these heritable IP components included HLA region genes, which is not surprising, but also other genes such as B2M and LAIR1. Since a large proportion of these heritable IP components were in the HLA region, we wanted to assess how much the HLA region was contributing to the phenotypic variation of these IP components. So we conducted a two-state GCTA analysis, one with just the HLA region and one with the rest of the genome excluding the HLA region. And so here in blue is the phenotypic variation explained by the HLA region, and then in orange, phenotypic variation explained by the rest of the genome. And so with this analysis, we were able to categorize 17 of our 137 heritable IP components where HLA regions the HLA region was contributing greatly to phenotypic variation, and 120 where the rest of the genome was contributing to the phenotypic variation. So for the 120 IP components outside of the HLA region, we did a genome-wide association analysis and identified 482 significant associations, of which 81% were cis associations and 19% were trans. On the right here are labeled some of the IP components that we detected significant associations for, including immunoinhibitors such as LRB2 and LAIR1, but also interleukins such as IL-2RA and IL-10RA. For the 17 IP components within the HLA region, we employed a separate pipeline because the HLA region has such complex LD structure. And so we conducted a conditional genome-wide association analysis, the premise of which is we conduct an association and identify the most significant hit. Then we rerun the association using that significant hit as a covariate, and then repeating this process until no more significant hits are remaining. So we did this analysis with HLA region expression quantified with traditional alignment to a reference genome, but also allele-specific expression with the patient-specific HLA alleles, and identified 50 significant HLA region associations. After identification of tumor immune microenvironment associated variants, we wanted to assess the cell type specificity of these variants. So first, we employed a similar pipeline to the GTEC consortium for detecting cell type interaction EQTLs. And the setup for this was doing a linear regression like we did in our discovery analysis, but adding an immune infiltrate term and also an interaction term, which included the genotype vector and the infiltrate vector. And so significant IEQTLs had a significant coefficient for this interaction term. Doing this analysis, we identified 36 significant M1 macrophage IEQTLs, 32 significant macrophage IEQTLs, and 21 significant dendritic cell IEQTLs. And so this suggests that a number of our tumor immune microenvironment associated variants might be specifically modulating macrophage and dendritic cell activity. Next, to assess the cell type specificity, we used the DICE database, which came from Vijay's lab at La Jolla Institute for Immunology. And what they did is they did cis-EQTL profiling of 15 immune cell subsets from 91 donors. And they were able to identify these immune cell specific EQTLs, one of which is shown here. This is an EQTL for IRAK3, specifically in naive B cells, but not in other immune cell subsets. So of our tumor immune microenvironment associated variants identified through the Tcga analysis, not surprisingly, we identified a monocytic EQTL associated with LILRA3 expression, and this is consistent with our IEQTL analysis, where we found a large proportion of our tumor immune microenvironment associated variants to be acting through macrophages or monocytic lineages. Overall, of our 475 tumor immune microenvironment associated variants, a large proportion were Th17 EQTLs, classical monocyte EQTLs, and Th2 EQTLs. So after assessing the cell type specificity of these variants, we wanted to see their role in immunotherapy response. Immunotherapy is a promising therapeutic option, but response rates remain low, and we need better prognostic biomarkers. So we wanted to assess whether germline variants could serve as prognostic biomarkers. So we started off by collecting 6 publicly available immune checkpoint blockade trial cohorts with normal whole exome sequencing and RNA sequencing available. Most were melanoma cohorts, but we also had a non-small cell lung cancer cohort and a renal cell carcinoma cohort. And then using optimized classifiers with the Optuna package, we evaluated the predictive value of our tumor immune microenvironment microenvironment associated variants in these cohorts, and we're able to achieve an AUC of at least 0.65 across all cancers using just germline variation. So with these promising results, we wanted to identify variants that were significantly associated with ICB response. And so we did a meta-analysis with our 6 ICB cohorts for which we ran associations with responder status. And we identified 23 variants significantly associated with ICB response. And here we are showing the behavior of these variants in the context of responder status amongst all 6 cohorts, IP component expression, immune checkpoint expression, and immune infiltration. And so as we know, variants have very complex effects. And so this is a very limited view of how the variants could be behaving in ICB patients. However, one thing that stuck out to us was the number of macrophage-related genes that these ICB significant variants implicated. And so we wanted to see if these 23 variants were associated with macrophage infiltration in ICB patients. And so we built a burden score where we took the 23 variants, oriented alleles such that they were associated with higher odds of being a responder. And what we found is that individuals with larger burden scores had higher M1 macrophage infiltration compared to M2 macrophage infiltration. And so this suggests that the tumor immune microenvironment associated variants might be affecting ICB response through modulation of the myeloid landscape. And indeed, a lot of the genes that the ICB variants implicate are biologically relevant to immune response, such as LILRB2, for which actually there is a small molecule inhibitor in clinical trials right now to be used as a single agent or in combination with pembrolizumab, and it has promising results. CTSS is another gene which has a role in antigen processing and T cell activity. And LAIR1 has a role in CD8 positive T cell exhaustion through interaction with collagen. And so, since a number of these genes seem to have a role in immune response, we wanted to validate them in murine models. And so, in collaboration with the Gutkin lab, and all of these experiments were done by Vicky Wu from the Gutkin lab, we implanted MC38 cells in mice and then treated with anti-PD-1 and did qPCR and found a significant upregulation of CTSS and CTSW in responders compared to non-responders. Interestingly, this was post-treatment, and these are immunoinhibitors. So this touches on an observation that we have made where immunoinhibitors seem to be upregulated post-treatment, but if they have decreased expression pre-treatment, this leads to better responses. And so we wanted to test the other side, the flip side of this coin, to see if we inhibit CTSS, do we see better responses and slowed tumor growth. And indeed, when we treat with anti-PD-1 and anti-CTSS and the combination, we do observe slowed growth of tumor cells in the combination treatment compared to anti-CTSS treatment and anti-PD-1 treatment alone and compared to the control. And so this is promising, and it's still preliminary, but it suggests that some of the genes that our variants are implicating could also have therapeutic implications, which is very exciting. And so to conclude, we ran a comprehensive analysis of germline variants modulating the tumor immune microenvironment and identified 482 significant associations outside of the HLA region and 50 within the HLA region, and found that they specifically modify macrophage and dendritic cell behavior. We assessed the predictive value and found they were predictive of ICB response through modulation of the myeloid landscape. And lastly, we experimentally validated a number of genes implicated by our tumor immune microenvironment associated variants and found them to be differentially expressed in mouse anti-PD-1 responders versus non-responders, and have promising preliminary results of anti-CTSS as a potential therapeutic add-on. And so with that, I'd like to thank both of my PIs, Hannah and also Jill, who have been really helpful with not only this project, but also other projects that I have done over my PhD. And also I want to thank the whole Carter and Meseroff labs, who have been amazing, not just on this project, but other projects. And also thank the VJ lab at LJI and the Gutkin lab, specifically Vicky, who has been really helpful with the experimental validation of these genes. And lastly, want to thank my funding resources. And so with that, I will take any questions that there might be. Thank you for listening.